Okay, so my name is Jordan Von Eggers. I'm a graduate student at the University of Wyoming. I'm in the program in ecology, and I'm advised by Doctors Brian Schumann and Dr. Amy Christ. And I grew up in the Seattle area and did my undergrad at the University of Washington. So it's been really great to hear all the talks today about all the research that's going on in Washington. And so I just want to take a second to thank the organizers for putting on this conference. So today I'm going to talk about how we can use lake sediments to track plankton biodiversity throughout the Anthropocene. So as I was scrolling through my field photos, I came across this picture of Monogram Lake in North Cascades National Park. And I think this is a great follow-up to Dr. Walsh's talk, because I also think it kind of symbolizes the spirit of this project in a snapshot. And this is because we often think of these mountain lakes as these really pristine ecosystems, really nestled away. But as you can see in this photo, human impacts, for example, increasing wildfire due to climate change, can really reach even these remote spots. But the fact that these lakes are relatively undisturbed compared to their lower elevation counterparts really makes these mountain lake ecosystems useful as sentinels of change. And mountain lakes connect to human activities through atmospheric processes, so changing climates and nutrient loads, and also via this little helicopter dropping fish off here. So mountain lakes are generally cold, they possess few nutrients, and therefore really have low productivity, and they're also naturally fishless. Yet today we're really transforming these lakes through introduction of non-native trout, or through atmospheric nitrogen deposition, and warming temperatures. So fishless lakes are host to really high densities of insects and large zooplankton, and large zooplankton in particular can outcompete smaller zooplankton for phytoplankton resources. And so this can manifest as our clear oligotrophic lakes. And to show you what one of these zooplankton look like, here's a video of this large red copepod at the top of one of these cores. And hopefully my video works. So these are actually really the top predators in the open water system because they're really efficient swimmers. They're really effective at eating phytoplankton, and they're actually the only zooplankton that eat other zooplankton. Okay, so all mountain lakes were naturally fishless, yet today across the western United States, 60% of all mountain lakes and 95% of those deeper, larger lakes now contain trout. And fish can have really drastic and long-lasting impacts on lake ecology because essentially they can induce trophic cascades. And so ultimately, after fish are introduced, we see both increases in small zooplankton and phytoplankton. And I'll just mention that these effects don't just stay within a lake. Introduced fish have been associated with declines in amphibian populations, alpine nesting birds, snakes, and even spiders. Okay, so our next stressor is atmospheric nitrogen deposition. So here's a map of total nitrogen across the western United States. And this atmospheric deposition comes from industrialization and agriculture. And I'll play this for you again. So when you look at this GIF, note that this isn't a topographic map, but we're seeing nitrogen accumulating in the mountain ranges. And this is due to those winter months where there's lots of precipitation that's falling on the mountains as snow. And so generally these high alpine lakes are in settings that have low nutrients in general. And so this makes them oligotrophic, but now they're getting pumped with nitrogen. And these nutrients really have consequences for overall primary productivity, but also they can change the composition of the phytoplankton communities as well. And so then our third and final stressor is climate change. So we know temperatures are warming across the United States, but I think this map nicely shows that some places are warming quicker than others. And climate change can affect organisms and communities by modifying the metabolic rates of primary producers, altering the growing and reproductive season of different organisms, which we've heard some talks about, and other phenomena that can kind of propagate throughout the ecosystem. And there's more than just these three threats to the mountain lakes, but in my project, we're just trying to kind of isolate these three effects. Okay, so we just had a quick overview of the three stressors. And I think this next figure really sums it up nicely because this figure shows how natural lake settings, so in the yellow and gray boxes, and anthropogenic stressors in that purple box, all influence various organisms within a lake. And then the DNA from all of these organisms sinks to the bottom of the lake and accumulates in the sediments. And so this is the main tool I'm using during my PhD is sediment cores, and I'm representing it with this orange bar here. And so lake sediments can provide this really useful tool to have, because they record evidence of both the conditions and the communities from the times that they were deposited. So we can use the cores and specifically the DNA in them to see what existed back then to kind of establish baseline conditions and then see how those communities can change through time. So this leads to our primary question, which is how are these originally fishless lakes being influenced by these three stressors? And to show you what some of the outcomes could be potentially, let's say fish introduction was really important for changing lake communities. Maybe we'd see that our lakes kind of cluster as like fish and fishless lakes in the present. But maybe things like regional factors and global factors like climate change and nitrogen deposition are really important for these communities. Maybe we would see that they change kind of in a similar way throughout time. Okay, so to answer this question, I collected 14 sediment cores, 7 with fish introduced and 7 without fish introduced in Washington, California, and Wyoming. And we chose these sites because we really wanted to capture different degrees of warming and nitrogen deposition. And so California here has higher rates of warming and deposition compared to both Washington and Wyoming. And I also selected these regions because the national parks in Washington and California have really detailed stocking records. They've got long-term lake monitoring efforts, and they have sometimes extensive removal projects to help with some of the removal projects of fish to help with some of the declining amphibian populations. So here's what coring and subsampling looks like. It's a little less intensive than Dr. Walsh's sampling, thankfully. And I've listed all of our methods we're using here, but the two important ones that we're going to talk about today is I've dated the sediments using lead-210, and our biological response, we're using DNA metabarcoding. And so we're looking at eukaryotic plankton. So this includes both phytoplankton and zooplankton, like I showed you in that video earlier. And so far we have dates for about half of the cores, but we've got DNA for all of them. So I'm going to show just 2 preliminary results from what we have so far. Okay, so here we have an NMDS plot, and I'm going to put up all the DNA results from the plankton communities from the 14 cores. But first I want to explain how this plot works. So here's an example of 2 samples or 2 plankton communities that are from different lakes that are pretty close together. So we would say that they're similar to one another. But if 2 samples are farther apart, we'd say that these communities are more dissimilar from one another. Okay, so first I'm going to just put up one lake. I'm just going to put up Canyon Lake. So here we have one full lake sediment core with the larger points on the right indicating historic communities, and then smaller points on the left indicating more modern communities. And so what we see is that the historic communities from this lake are all kind of similar to one another, but we see that modern communities become more dissimilar from the historic communities as we go from the right side to the left side. So now I'm going to add in the rest of the lakes, but I'll add them in by the state. So here we have cores from Wyoming, California, and Washington. And if you remember, we really want to compare cores from lakes with and without fish introductions. And so if we think that fish are changing plankton communities in a similar way, we would expect them to cluster by groups of fish and fishless lakes, just like we talked about earlier. And so what I've done here is just outlined all the cores that have had fish introduced, but we only have dates for half of them, so we really only know for about half when the fish were actually introduced. So, but I've outlined the whole lake core, but in reality, all the historic communities will be fishless. And so what we're seeing is that these lakes really aren't clustering by fish and fishless, and so perhaps fish isn't the primary driver of plankton communities overall. But what we do see is that there's lots of variation in historic communities, and then we see this kind of convergence in more modern communities, potentially suggesting some homogenization. So this makes us think that perhaps it's these global and regional signals, so nitrogen deposition and climate, that are really driving the communities. Okay, and then I just have one last result to show briefly because I know I'm running out of time. So while we don't think that fish may be really important for the whole community, since fish eat zooplankton, maybe it's really just important for zooplankton communities. So here I have 2 lakes, one that's had fish introduced, one that hasn't. And they have, these are cores that have dates, and they're from Wyoming. So when we look at the record for the DNA of just zooplankton in the fishless lake, we see that that big red copepod that I showed you a video of dominates throughout the entire record. Whereas the lake with fish has some variability in the past, but potentially fish are changing those zooplankton dynamics after they've been introduced. So just to conclude, There's many threats to mountain lakes. Sediment cores can be really useful to track those. We think that regional and global factors may be driving some of these recent changes, but maybe fish are important for zooplankton. And with that, I'll say thank you to my advisors, committee members, and I'd love to thank people at the National Park and Forest Service, especially Rebecca Lofgren and Ashley Rawhauser, who helped me pick out where to core. And I want to thank my funding sources and field assistants. With that, I'll take any questions. Let me find the box. Okay. Let's see.
Megan, you're muted.
Sorry, such a rookie mistake. Thank you very much for your presentation, Jordan. Again, please put your questions in the chat or raise your virtual hand. Megan says, great job, Jordan. I'm glad you're working with Brian. And Robert has a question. He says, did you look at phytoplankton productivity or turbidity?
Yeah, I haven't looked at them yet, but we are planning to look at biogenic silica as a measure for essentially primary productivity. I mean, it's just looking at essentially the silica from diatoms that we might be able to kind of gauge that. But I think that a lot of the communities, especially in the Rocky Mountains, are dominated by diatoms. So that can be kind of a proxy for overall like productivity by looking at that biogenic silica that's in the core. So, but I haven't looked at turbidity. I'm not sure. Yeah, it'd be interesting to see if there's a way I could reconstruct that. But I'm curious if you have any suggestions.
John says, great job when referring to historic populations. What general range of time does this refer to? How far back in time do you think these records can represent?
Yeah, it's hard because the method we're using for dating is really good from like 1850 on. Otherwise, we'd need some sort of charcoal at the bottom to kind of connect our ages. But if we kind of just extrapolate back from the sedimentation rates through lead 210, this kind of can range from anywhere maybe like 300 to maybe 500 years. But it can be kind of hard to get radiocarbon dates if they're really close, like within the last few hundred years. Sometimes radiocarbon isn't that helpful. So we generally think it's a few hundred years. Oh yeah, pigment types. That's an interesting thing. We hope to look into that.
All right, excellent. Well, thank you very much, Jordan.